Hello everyone, today I'm going to go through the steps of cellular respiration. Cellular respiration begins with glycolysis. And glycolysis means splitting sugar. This takes place in the cytoplasm of all cells. This begins with glucose, which is a 6-carbon sugar. Glucose is then split in half into two 3-carbon molecules. But to do this takes a little bit of energy. So 1 ATP is converted to ADP for both sides. Now this 3-carbon molecule undergoes a series of enzyme reactions which convert one NAD to NADH and one ADP to ATP on each side. And it ends with the enzyme kinase. Kinase takes a resulting molecule of PEP plus ADP, and it's going to convert that into 1 ATP plus pyruvate, which is a 3-carbon molecule still. We do that on both sides. And at this point, our yield up to here, up to this point here, is net of 2 ATP two NADH and 2 pyruvate. Now the ATP can go do work in our cell. The NADH we're going to save for our electron transport chain. And that gives us pyruvate, which can either go to the mitochondria to finish cellular respiration. Fix that R. There we go. Or it can go to fermentation. Now that's the same for both pyruvates. So this one could go here or here as well. For fermentation, we have 2 different types. We have alcohol fermentation and lactic acid fermentation. Okay, now fermentation all takes place in the cytoplasm still. And it is anaerobic. So that means no oxygen. Okay, so these can take place. This one here takes place in our prokaryotic cells only. This could be prokaryotic or eukaryotic cells. Lactic acid, that's when you get those muscle pains and strains when you've been working too much, when you've been writing all of this and you need to shake your hand out because it's sore. That's lactic acid. And alcohol, you guys are familiar with. Alcohol fermentation is how things turn. Let's see. We make wine from grapes. We make beer from barley and hops. All those alcohols are created here. Lactic acid is also how we get cheese. So we can see this in both animals and some of our prokaryotics. Alcohol only in our prokaryotics. All right, that means that I have pyruvate going to the mitochondria. In order for it to be useful in the mitochondria, we have to first oxidize our pyruvate. So our next step is oxidation of pyruvate. Now, oxidation of pyruvate, this happens only in eukaryotic cells. Okay, it has to be a eukaryotic cell, and it takes place on the proteins as it moves through the mitochondrial membranes. So it's as moves through our mitomembranes. And it's a pretty simple process, but it's really important. Okay, so we start with our pyruvate, which is a 3-carbon molecule. And we are going to go through our mitochondrial membrane. So we have our outer membrane, and we have our folded inner membrane. And as it travels through, we're going to pull off a carbon, giving us a 2-carbon molecule. But that carbon has to have something to catch it. So we combine it with some oxygen. We get carbon dioxide, and then this can leave our body as we exhale. But every time we convert a carbon or a strip of carbon to make carbon dioxide, we convert one NAD to NADH. Okay, now this 2-carbon molecule here at the end, this is called acetyl-CoA. And this is in our matrix. Okay, so oxidation of pyruvate takes place as we move through. Now remember that we had 2 pyruvates, so all of this is times 2 for each glucose. Okay, so all of these are times 2. That means I get 2 acetyl-CoAs as well. So as we go through this, our yield is going to be 2 carbon dioxide, 2 NADH, and 2 acetyl-CoA. Now these we breathe out. NADH we save for the ETC. And acetyl-CoA moves on to our Krebs or citric acid cycle. Okay, so let's take this acetyl-CoA and send it to our citric acid cycle. So the citric acid cycle is also known as the Krebs cycle, after Hans Krebs. Okay, this takes place only in eukaryotes. because it takes place in the mitochondrial matrix. Okay, now remember, that's the inside of our mitochondria. Okay, so the matrix is inside here. And that's where we're going to be looking at the citric acid cycle taking place, is inside here. So acetyl-CoA is a 2-carbon sugar. Okay, it came from oxidation of pyruvate. And with this, one of our key things to remember is that there are 8 steps. And remember 6, 6, 5, and then 4s. Okay, so there's 8 steps. I'm going to go 6, 6, 5, 4. That means 4 more 4s. 1, 2, 3, 4. And these are all carbons. So I'm going to put my carbons in here. All right, so this acetyl-CoA is going to combine with our 4-carbon molecule to give us a 6-carbon, and that is called citrate. Okay, or citric acid, which is how we get our name, citric acid cycle. But this isn't in a form that we can really do much with, so we're going to rearrange it. And then we're going to go from 5 to 6. As we go from 5 to 6, we have to leave a carbon behind. So that goes out as CO2. But remember, every time we convert CO2, we get to convert an NAD to NADH. We're going to then remove a carbon again for this next step. So CO2, NAD is converted to NADH. We've done an awful lot of work here, so we're going to get a little reward in the form of changing one ADP to an ATP. And then we're going to rearrange this, but again, we've done some work and we're going to get a little bit of energy out of this. Fix that so you can tell that's a 4. There we go. But ATP is the really good energy. We're going to do a storage form here. And we're going to change an FAD to an FADH2. Now we're going to rearrange this. And then we rearrange it one more time to get it ready to do the cycle again. But we're going to get a little bit of a bonus here. NAD. Make that actually look like a D. Is converted to NADH. Now remember that when we went through oxidation of pyruvate, we wound up with 2 acetyl-CoAs. So all this has to happen 2 times. Now here, if we look at the yield, okay, 1 turn is going to give us 1 ATP, 1 FADH2, and 3 NADH. That means that 2 turns is going to give me 2 ATP, 2 FADH2, and 3 NADH. Oh, nope. Forgot to multiply that by 2. 6 NADH. Okay, now if I add everything together so far, So 1 glucose would give me up to this point a total of 4 ATP that can go do work, 10 NADH, and 2 FADH2. Now these are going to go to our electron transport chain. Okay, so these go do work. These are now stored to give us energy in the electron transport chain. Now in the electron transport chain, we have our inner mitochondrial membrane, so the cristae. So we have our outer membrane out here, and then we have our like folded inner membrane, and we're going to give it a big fold so we have room to work. And on this membrane, we have a series of proteins. And our last protein here is going to be ATP synthase. Now, in our matrix here, we have created a whole bunch of NADH and FADH2. Now, the hydrogen here at the end, okay, if we look at what hydrogen is a proton plus an electron. What we're going to do is start stripping these and separating that. So I'm going to have an NADH that gives its hydrogen to a protein, and I get NAD. In here, we're going to separate that hydrogen and electron, and my hydrogen is going to get kicked out into here. My electron moves along and gets a new hydrogen, which gets kicked out. And then my electron moves along to the next one, where it gets a hydrogen, and then kicks that one out as well. So now I'm creating all these hydrogen ions in this space with less here. That gives us a chemical gradient. So more here, less here. Now, because of this, these hydrogens are building up. They want to be able to spread back out. Okay. Every time we have a hydrogen wanting to spread out, it can go through our ATP synthase into our matrix. And as it does that, it converts an ADP into ATP. So 1 NADH is able to give us 3. Okay? 3 ATP. But we still have a problem of this electron here. Now this electron has to be accepted by something. Otherwise, this whole like water slide is going to get all backed up and they can't get out. So this electron is accepted by oxygen. Okay? And specifically, we give it half an O2. Make that a little bit bigger so it's easier to see. O2, there we go. And it's going to be a half of an O2 plus 2 hydrogen, because we have hydrogen all over the place, plus this electron, and that's going to give me water. Okay. Now, FADH2 is always a little late to the party, okay, and it can't start here. Our FADH2 is going to give away hydrogen, and it's going to actually start here. Okay, so every FADH2 can give me 2 ATP. Okay, so it's going to pump more through. They're going to come back. We wind up having the FADH2 entering at the second protein. Okay. Now from this, if we look at, we started with 10 NADH, so 10 NADH. That's from glycolysis, oxidation of pyruvate, and Krebs cycle. Each one gives me 3 ATP. That gives me a total of 30 ATP. We add 2 FADH2 from our Krebs cycle, and those each give me 2 ATP, giving me 4 ATP. And then in glycolysis and Krebs cycle, we were able to produce from Krebs plus we had 4. So we're looking at about 38 ATP. Now you'll see anywhere from 36 to 40 when it's asked how much ATP is produced. Okay, so 38 right there. Sometimes these hydrogens get used before they come back through. Sometimes they kind of just float around here. So our average is about 38 ATP. Okay, now I highly recommend that you take a look at the drawings that I've posted to give you a way more detailed and understandable idea of each of these steps. Okay, just a quick view of those. Here's glycolysis. Here's our oxidation of pyruvate. Our citric acid cycle. And electron transport. Okay, so if you have questions, please let me know. Bye.